Hi, I'm Diana Friedman with Research to Prevent Blindness.
And I'm Matthew Levine with the American Macular Degeneration Foundation.
And we are joined today by Dr. Claudio Punzo, who is the Associate Professor and Research Vice Chair at UMass Chan Medical School, where he conducts research to develop treatments for photoreceptor degenerative diseases such as age-related macular degeneration. And Dr. Punzo recently received a Research to Prevent Blindness American Macular Degeneration Foundation Catalyst Award for innovative research approaches for age-related macular degeneration. So we'll be asking Dr. Punzo a few questions today about his work. Welcome, Dr. Punzo. So congratulations on your award. What will this award in a general sense enable you to study?
Well, thank you very much. And first, I would like to thank Research to Prevent Blindness and the American Macular Degeneration Foundation for the award. The award will enable us to basically develop a therapy for the wet form of age-related macular degeneration and also set proof of concept that the kind of approach that we are taking may be applicable to more diseases than only wet age-related macular degeneration.
And why is this topic so important?
The topic is important because wet age-related macular degeneration affects a large number of people that are 65 and older. It is a very severe condition that leads to blindness, it leads to central vision loss, and it's really debilitating. And the current treatments are not ideal. They work very well, but they are not ideal because patients have to come very often to the clinic to receive injections, and this treatment would be much better.
So how long have you worked in this area?
My lab has been conducting research on age-related macular degeneration for the past 7 years approximately, and we have started to develop also treatments for wet age-related macular degeneration in the last 3 years.
So the idea of gene therapy holds exciting promise for those with macular degeneration and other gene-related eye diseases, but gene therapy is a term loosely applied to several approaches. What are the main gene therapy approaches that are under investigation as new treatments for AMD specifically?
So there are gene therapy approaches for the wet form of AMD, the one that had also been funded. And then there are treatment approaches for the dry form of AMD. Most of the treatment approaches for the dry form are actually, gene therapeutic approaches are actually around the complement system, increasing expression of factors that inhibit the complement system. So you basically would inject the virus and then hope that the protein that is secreted by the cells after the infection would prevent progression of the dry form, which is generally referred as geographic atrophy. For the wet form, there are gene therapy approaches that basically mimic what is done with the injections, the intravitreal injections that are currently used as treatment. There, normally a protein is injected that inhibits the vascular endothelial growth factor to promote vascular growth. And these gene therapies basically would transduce cells that then express that protein and secrete that protein and would produce it endogenously in the eye. This would avoid repeat injections that patients have to do at the moment.
Wonderful, and I'll let Matthew take it away with some questions.
Thanks. Dr. Punzo, obviously, you've described several different approaches to gene therapies, and I guess in my understanding, there's ways of replacing faulty genes, there's ways of correcting faulty genes, and then there's using gene therapy to make the cells in the eye itself manufacture a treatment drug. Could you sort of talk to our audience, which is mostly non-scientists, about this siRNA therapy that you're developing and how it differs from the other approaches?
So with our therapy, we manipulate as well gene expression, but we do not introduce a gene. we basically introduce a very small molecule that diffuses into cells and then reduces the expression of the endogenous target gene that we are targeting. So the sequence can be designed gene specific. It's a very short sequence of about 20 base pair. And because the molecule is small, it just diffuses to the back of the eye and targets all cells. So with our therapy, we can very well control one specific gene and reduce the expression level of that gene, but we cannot design it in a way that it goes specifically into one cell type. With a gene therapy, you can often design the expression of the gene to be cell type specific, although the virus may infect multiple cell types. The advantage of our therapy is that it can be turned off, meaning you do an injection and eventually the drug fades away, whereas a gene therapy is generally permanent. So if there are side effects, you cannot really turn it off anymore. The advantage of the gene therapy would be that it is permanent, but it is actually much more difficult to target a lot of cells with the virus because viruses don't travel very far when you inject them in the eye to the back of the eye. And you would have to do a subretinal injections, which generally with a gene therapy is much more invasive as a procedure versus the intravitreal injection that we do with the siRNA, which is not non-invasive.
So that's the intravitreal injection. That's similar to what people are getting with the anti-VEGF therapy. So it could be performed very easily in a doctor's office, I'm assuming, as opposed to maybe the subretinal, which sounds like more of a hospital procedure. Is that right?
That is correct. The subretinal injection is considered more of a surgical procedure and is done in a hospital setting, whereas the intravitreal injection can be done by any ophthalmologist in the regular eye clinic. Okay, so it seems that one real clear advantage of the therapeutic approach you're taking is that it's easy to administer. It is, yes, yes.
Yeah, but in the application you wrote for the grant, you also talked about other areas where this is an advantageous approach to therapy. I think you mentioned cost effectiveness, but also durability of the product and the fact that you might be able to deliver this to remote regions in the world. It sounds like a product that could eventually have a lot of advantages. Could you speak about that a little bit?
Yeah, so the current intravitreal injections with anti-VEGF proteins have the problem that it is not long-lived. So they last about 1 to 3 months at maximum. Because it is a protein, it needs to be produced in a cell line, so in a cell culture. Each batch needs to be tested for pathogens and other infectious diseases. So it's much more costly to produce. The drug is much more expensive. Our drug is basically a chemical compound that can be synthesized, can be sterilized very easy, produced in large batches, and is very stable compared to a protein. So it can be shipped off in far remote regions of the world. It can be used there. It doesn't need necessarily to be cooled. And the advantage of our drug is that it seems to last currently, from our knowledge, at least 6 months. We believe that it might be even possible to do injections at the frequency of once every 9 months to once a year. And this would obviously dramatically reduce the burden on patients that have to go in at a very high frequency, because we are talking here of elderly individuals that often lack the means of transportation, or if you're talking of remote regions, these patients can't show up to the eye clinic at a high frequency. So this would be one of the big advantages. Cost-wise, our drug can be produced chemically at about $1 per eye, which is significantly cheaper than the current drugs.
It's fantastic. So you've alluded to this before. Just let's say there is another treatment that's developed, a gene therapy approach whereby cells in the eye are induced to produce a medicine that's permanent, it's ongoing. But you seem to indicate that the siRNA approach might have advantages because you might not really want to have that drug produced forever in the eye. Could you speak about that?
Yeah, so there is concern in the field that permanent knockdown of VEGF has negative consequences because it's required for maintaining proper vasculature permeability and health. And the current treatments, patients receive an injection, and so far the side effects have been minimum because the amounts that stay in the eye after the injection of the protein are very low. Once you produce it endogenously in the eye, there could be severe side effects over time because you have a much higher level that is constantly present. The siRNA lends itself also much more to regulating how much knockdown you do because you can adjust the dose very easily. Whereas once you inject the virus, you know, the cells that you infect are the cells that you infect and you constantly produce a certain amount. You can't recorrect it. We could, you know, if the dose is too high in our case, we could in the next injection lower the dose, make it half as big. So you can correct each patient individually depending on how the patient responds. Whereas once you have the viral gene therapy, there is no correction possible after that.
So we're talking about therapy for macular degeneration, but what other eye conditions could this same approach be applied to?
So specifically, our therapy could be definitely, well, I don't want to say definitely, but could also work for diabetic retinopathy. The siRNA approach in itself lends itself to most mutations that are dominant negative in the eye and cause retinitis pigmentosa. So all forms of dominant retinitis pigmentosa could be easily treated with the same approach. We basically would just have to design a different sequence and maybe change the chemistry a little bit. We have identified different chemistries for different cell types, or that at least enrich in different cell types. And then we could basically target the photoreceptors more efficiently and thereby develop therapies for the dominant negative forms of retinitis pigmentosa.
Well, thank you for explaining, you know, the science behind this. I'm going to turn it back over to Diana. I think she's got some other questions.
Yeah, you know, one question that we just chatted about very briefly before this interview was your timeline. What does the ideal timeline look like for this kind of work?
Well, we hope that, you know, for this approach, we can basically identify a working target sequence within a year and a half, and then start to do the animal work in the next year and a half, and get enough preliminary data to show that conceptually it works. You know, the approach is on paper, in concept, very logical. That's often the case in science. And then, you know, once you try to translate it and get it to work, suddenly you find that certain things don't work out the way you thought they should work out. Right. But we are very hopeful that this is going to work.
Yeah. And of course, that's why RPB and AMDF have stepped in to provide this funding, because we think it's really important for you to take that next step. What has the role of nonprofits been in your research career to date, and the type of support that they've given you?
They actually have played a very important role in my career. You know, I have very often initiated projects that sound sometimes crazy, and it's very difficult to get funding from the NIH for ideas that are kind of strange initially, and private foundations are a very good source of funding for these kind of ideas, because they often provide that initial step of money that you need to get enough preliminary data and then apply for funding from the National Eye Institute.
Right. Yeah, and you know, private foundations, obviously, and nonprofits have the ability to fund research that's a little more high risk, high reward. So we see that as a really important role that we fill in taking new ideas and actually bringing them to life in a way that perhaps some other funding sources cannot. But it really helps to drive innovation. So we're really, both organizations are happy to be in that role. So what would you say excites you most as you embark on this research project?
What excites me most is being able to get it to work and show that it works. Because if we get this as a proof of concept to work, I think it opens the field for many more treatments that I've mentioned before, and would really change the way we treat eye diseases. The siRNA has really a huge potential for eye diseases because it's very simple, it's very straightforward, it's cheap, and it's very modular. It's kind of like a Lego kit, right? Once you have determined the chemistry that gets best into a certain cell type, you just change the sequence. It may allow us also to reduce the need of repeat clinical trials, right? Or expensive clinical trials, make them much smaller and shorter, because you're basically just changing a sequence on the same molecule. Once you've shown that the molecule itself is not toxic, changing the sequence would not necessarily change toxicity. And if the gene is anyway, let's say photoreceptor specific, you don't really worry of any off-target effects in other cell types because these cells don't have that gene, right? So you basically, if we get it to work for the anti-VEGF and if we get it to work for one photoreceptor gene, we can basically just, you know, play around and change the sequence for all the other genes that need to be treated, right? And that goes very fast afterwards and will allow to develop therapies for diseases that may be more rare, where few people have access in a very cheap way.
Well, it's very exciting, and we wish you the best of luck with your work, and we look forward to hearing more about it. I know you said you'll be looking to publish some papers later this year or early next year. We will certainly share those with our audiences to see your progress. And thank you so much for taking the time to describe your research to us today. We really appreciate it.
Well, thank you very much for the opportunity to describe my research, and I would like to thank you both again for funding the research.
Thank you.